Hello everyone and welcome to vEgyan Ghar's YouTube channel. Today we shall discuss the 2019 TIFR GS paper. TIFR GS is a nationwide entrance exam conducted by Tata Institute of Fundamental Research for admissions in postgraduate courses offered by it. The paper has 4 sections with 15 questions each. The sections are General Science, Physics, Chemistry and Biology. In this video, we shall discuss chemistry and biology sections of the paper. Here is the chemistry section. Element Y has a nucleon number 19 and a proton number of 9. Which group in the periodic table does it belong to? A. Group 1, B. Group 3, C. Group 7 or D. Group 8. The number of protons in the element Y are 9. So the number of electrons is also 9. So the electronic configuration is 1s2, 2s2 and 2p5. So the last shell, that is the second shell, has 7 electrons. So the number of electrons in the outermost shell of the element in a group is the same as the group number. Therefore, correct answer is option C. Which statement is true for the following chemical reactions? CuCO3 plus heat gives CuO plus CO2. CuO plus SNO gives Cu plus SNO2. A. CO2 is oxidized and SNO2 is reduced. B. CuCO3 is oxidized and CuO is reduced. C. CuO is oxidized and SNO is reduced. D. SNO is oxidized and CuO is reduced. Now, we can understand this question by writing the oxidation states for the molecules in the second reaction. These are as follows. In the second reaction, the oxidation state of Cu decreases from +2 to 0. So as you can see, this is decreasing from +2 to 0. The decrease in oxidation state signifies reduction. Similarly, the oxidation state of Sn increases from +2 to +4. So SNO is oxidized. So the correct answer is option D. SNO is oxidized and CuO is reduced. Dinitrogen tetroxide N2O4 breaks down into nitrogen dioxide NO2. If the reaction is reversible and endothermic, which condition will give the largest yield of NO2? A. High temperature and high pressure. B. High temperature and low pressure. C. Low temperature and high pressure. Or D. Low temperature and low pressure. As the reaction is endothermic, heat is absorbed in the forward reaction. So we can expect that a decrease in temperature in this system will cause the reaction to shift in the reverse. That is, it should favor the exothermic reaction. Thus, an increase in the temperature will cause a forward shift to absorb heat in an attempt to restore the original conditions of the equilibrium. Similarly, low pressure will favor the forward reaction. So the correct answer is option B. Which of the following molecules will have a dipole moment? CH4, NH3, CCl4 or CO2? A dipole moment is the turning force a fixed charge has in a dipole in the molecule. The moment depends upon charge at one end of the dipole and its distance from the charge at the other end of the dipole. That is separation of charges. It is given by mu is equal to Q into D, where Q is the charge and D is the distance. That is separation distance. In more complex molecules with polar covalent bonds, the three-dimensional geometry and the compound symmetry determine whether there is a net dipole moment. Mathematically, dipole moments are vectors, so they possess both magnitude and a direction. The dipole moment of a molecule is therefore the vector sum of the dipole moments of the individual bonds in the molecule. Now, if we look here, both CH4 and CCl3 have perfect tetrahedral geometry, with their central carbons having sp3 hybridization. So they have zero dipole moment. Similarly, CO2 has a linear structure in which the direction of the two bond dipoles cancel each other. So the electron cloud is pulled in the opposite directions since oxygen is more electronegative, and the charges cancel each other. So it has a zero dipole. NH3 on the other hand has a lone pair of electrons on the N atom which pulls the electron cloud away from the H atoms. So it has a net dipole moment. So the correct answer is option B, NH3. When a non-volatile solute is dissolved in a pure solvent, the difference between the freezing points of the solvent alone and the solution is A positive, B negative, C 0, or D none of the above. The properties of a solution that depend upon the solute particles are called colligative properties. They also include freezing point of the solution. When a non-volatile solute is dissolved in a pure solvent, the freezing point decreases. So the difference between the freezing point of the solvent alone and the solution will be positive. So the correct answer is A. A helium atom is heavier than a hydrogen molecule. At 298 K, the average kinetic energy of the helium atom is A. Twice as that of hydrogen molecule, B. Equal to that of hydrogen molecule, C. 4 times that of a hydrogen molecule, or D. Half that of a hydrogen molecule. Now, helium is a monoatomic gas, while hydrogen is diatomic. However, according to the kinetic theory of gases, the kinetic energy of gases is given as Ke is equal to 3 kT by 2. Here, K is 1.38 into 10 raised to minus 23, and it is called as the Boltzmann constant. As you can see from this equation, the kinetic energy is independent of the molecular structure, but is dependent on temperature. So, kinetic energy of both gases will be the same. So, the correct answer is option B. Among the following, the one that gives positive iodoform test upon reaction with I2 and NaOH is molecule A, molecule B, molecule C, or molecule D. Iodoform test is used to detect a RCOCH3 or a RCHOHCH3 group. Positive test will give a yellow precipitate. Now, of these, option A and option D have terminal hydroxyl groups. So it is definitely not the answer. Here, option B has a CH3 group, a OH group attached to CH. So the answer is molecule B. So second option is correct. For an endothermic reaction where delta H represents the enthalpy of the reaction in kilojoule per mole, the minimum value for energy of activation will be A less than delta H, B 0, C more than delta H, or D equal to delta H. We can understand this question from a free energy diagram for an endothermic reaction. Here is the diagram. Now, from this diagram, we can see that delta H is positive. So, the products are less stable than the reactants. This is why the reactants have to absorb more energy to form products. So, the correct answer is more than delta H. That is option C. Here is the next question. Which of the following amino acids is commonly used in catalysis by enzyme? A. Alanine, B. Arginine, C. Serine, or D. Methionine. Enzyme active sites may have spatially arranged amino acids that are needed for catalysis. An example of these amino acids is serine. Some others include cysteine, histidine, asparagine, lysine, serine, threonine, tyrosine, etc. So here the correct option is serine, C. Which of the following compounds does not undergo ozonolysis reaction? A. 3-methyl-1-butene, B. 2,3-dimethylbutane, C. 2-methyl-2-butene, or D. 2-methyl-1-butene. Ozonolysis is a reaction that produces aldehydes or ketones from unsaturated hydrocarbons, that is having double or triple bonds, using ozone. Among these options, the option B here is a alkane. So it is a saturated compound. Therefore, it cannot be used to produce aldehydes or ketones. So the correct answer is option B. 4.8 grams of gas at 27 degrees Celsius occupied the same volume as 1 gram of hydrogen at 17 degrees Celsius and at the same pressure. What is the molecular weight of the gas? 0.1 gram, 0.48 gram, 5.2 gram or 9.9 gram? We know PV is equal to nRT, where N is the number of moles which is equal to given mass upon molecular mass. Temperature in degree Kelvin is equal to 273 plus temperature in degree Celsius. Now, in this question, pressure and volume are maintained at a constant. So, the only things varying are N and T. So, we can write as N1T1 is equal to N2T2. So, N1 can be calculated as 4.8 by X, which is given mass of gas, divided by molecular mass, which we need to find out, into 300, which is 273 plus 27, is equal to half, that is 1 gram of hydrogen divided by the molecular weight of hydrogen gas, which is 2, into 290, which is 273 plus 17 degrees Celsius. So from this we can calculate X is equal to 4.8 into 300 into 2 divided by 290, which is equal to 9.9 grams. So the correct answer is D. Consider a reversible reaction A gives B with forward and backward rate constants K plus is equal to K minus is equal to 1 second inverse. Suppose we start with 1 molar solution of A. How long will the concentration of A take to reach 0.75 molar? A 0.25 seconds, B natural log of 4 by 3, which is approximately equal to 0.29 seconds, C natural log of root 2, which is approximately equal to 0.35 seconds, or D, it will never reach that concentration. Now, from the question, it is important to note that the unit of the rate constant given is second inverse. So the reaction is a first order reaction. We can solve this like so. Minus dA by A, which is the rate, is equal to Kf A raised to m minus Kb B raised to n, where Kf and Kb are the rates of forward and the backward reaction respectively. Here, m is equal to n is equal to 1. So when we solve this, minus dA by A is equal to Kbb minus Kfa. Here we know that B is equal to A minus A naught. That is the final concentration of A minus the initial concentration of A. On solving this, we get minus dA by A is equal to A naught minus 2A. To find the time, we integrate dA by A naught minus 2A from A naught to A. On solving this, we get minus ln minus half ln A naught minus 2A from A naught to A. Substituting the values, we get the time is equal to minus half ln 0.5, which is approximately equal to 0.35 because it is natural log of root 2. So the correct answer is option C, natural log of root 2, which is approximately equal to 0.35 seconds. Which compound is most acidic? H2O, H2S, H2Se or NH3? Acidic compounds release H plus ions. Among the options, H2O is more acidic than NH3 as oxygen is more electronegative. Similarly, the bonds between H and Se are longer than that of H and S or H and O bonds. This is because Se has a larger atomic radius. So the acidity order from the highest to lowest will be H2Se, H2S, H2O, and then NH3. So the correct answer is option C. H2Se is the most acidic. Labeling of proteins with iodine is used for tracing. Which amino acid does iodine react with? A. Alanine, B. Aspartate, C. Lysine, or D. Tyrosine. Introduction of a radioactive iodine to certain amino acids is used for tracing. Iodine is introduced orthogonal to hydroxyl groups on amino acids. Only tyrosine in these above options has an hydroxyl group. So option D is correct. Now, you are given an oligonucleotide whose molecular weight is 10,000 Daltons. Assuming you want to make 100 μl of 100 μm stock, how much oligonucleotide would you need? A. 10 μg, B. 1 μg, C. 100 μg, or D. 1000 μg. 1 Dalton is equal to 1 gram per mole. We say this since one molecular hydrogen atom has mass 1 Dalton. So 10,000 Daltons will be equal to 10,000 grams, which is equal to 100 μg. So the correct option is C. Now we move on to the biology section of the paper. Here is the first question. Glucokinase phosphorylates glucose in the liver only when glucose levels are higher than normal. What property of glucokinase is responsible for this? A. High molecular weight, B. High Km, C. High Vmax, or D. The ability to act equally on D and L glucose. So glucose or hexokinase is present in liver and beta cells of pancreas. It is highly specific for glucose, but it is inactive at low blood glucose level. After a hearty meal, blood glucose level increases and insulin activates glucokinase to phosphorylate glucose. It is only possible if hexokinase 4 or glucokinase has a low affinity for glucose and needs higher concentration of the substrate to function. Lower the Km value, higher is the affinity of the enzyme. So the property of glucokinase responsible for this particular phenomena is its high Km value. So the correct answer is option B. Platelets contain A. Both genomic and mitochondrial DNA, B. Only mitochondrial DNA, C. Only genomic DNA, or D. No DNA. Platelets are membrane-bound cell fragments used in clotting. They are produced by the fragmentation of megakaryocytes, which literally translates to cell with a large nucleus. Platelets are produced in the bone marrow by the process of thrombopoiesis. In this process, they lose their nucleus. So, they contain only the mitochondrial DNA. So, the correct answer is option B. Protein X is composed of 700 amino acids. When resolved on a denaturing page, it shows a band corresponding to 130 kilodalton. The probable reason for such a discrepancy is A. Dimerization of the protein, B. Glycosylation of the protein, C. Phosphorylation of the protein, or D. Acetylation of the protein. In SDS page, molecules move according to their charge to mass ratio when electric current is applied. In SDS page, the detergent SDS is used to denature the protein from its native shape and open it into a linear piece. Glycosylation of proteins refers to the covalent bond formation between sugar molecules and specific amino acids in the protein. SDS doesn't interact similarly with these sugars, and thus glycosylated proteins may appear as a smear at the end of the electrophoretic run, while other proteins are resolved properly. So the correct answer is B, glycosylation of protein. A geneticist crossed two purebred, that is homozygous tall and short plants. In the F1 generation, she observed that all the offspring were of intermediate height. If she crosses one of the offspring with a short parent, what kind of progeny would you expect? Assume that the plant height is dictated by a single locus. A. All short. B. 50% tall and 50% short. C. 50% intermediate and 50% short. Or D. 50% intermediate, 25% tall and 25% short. The parental generation has homozygous characters for height. On crossing these two, we get an F1 generation with 100% intermediate height. That is heterozygous genotype. When this F1 generation with intermediate genotype, that is heterozygous, is crossed with short parent, that is homozygous recessive, we get 50% heterozygous genotype, that is intermediate height, and 50% homozygous recessive. Such a cross is called a test cross. Thus, the option C is correct. How would you test whether a phenotypic difference between two populations of a plant is due to environmentally induced plasticity or evolutionary adaptation? A. DNA sequencing. B. RNA sequencing. C. Grow both populations in different greenhouses. Or D. Grow both populations in the same greenhouse. Phenotypic plasticity refers to the ability to express varying phenotypes when exposed to different environment conditions. So, to check if the phenotypic difference is due to environment, we grow them in the same environment conditions, therefore the same greenhouse. So, the correct answer is option D. In an experiment, two fly populations are separately maintained for many generations. Population A contains closely related individuals. Population B contains a set of unrelated individuals. Over many generations, it is observed that the fitness of individuals in population A is lower than the fitness of those in population B. Why? A. Close relatives will not mate with one another. B. Close relatives compete for common resources more than unrelated individuals. C. The offspring of close relatives harbor more deleterious recessive mutations. Or D. Unrelated individuals are likely to contain new high fitness genes. Now, both the populations are maintained separately, so they do not interact. This is called a bottleneck effect. Population A has closely related individuals which have to mate amongst themselves. Thus, more deleterious mutations are observed in that population due to inbreeding depression, which leads to reduced fitness. So, the correct answer is option C. Of the graphs shown below, which is the most representative of the kinetics of ion transport through membrane channel? Graph A, B, C or D? The rate of transporter-mediated diffusion reaches a maximum of Vmax when the transporter is saturated. Therefore, graph D is the correct option. As you can see, it is reaching Vmax value after the saturation of the transporter. In case of simple diffusion, the ion concentration is directly proportional to the transport rate. That is graph A. So the correct option is graph D. Galvani discovered in the 1700s that frogs' legs jerked when touched with a piece of copper and a piece of iron simultaneously. This happens because action potentials are triggered due to which of the following properties of metals? A. Metals conduct charge away from the cell membranes. B. Soluble metal ions mimic neurotransmitters. C. The temperature difference between metals and tissues. Or D. The electrochemical potential difference between two metals. Galvani was the pioneer of electrophysiology. He stated that the jerking of frog legs was due to a type of electrical fluid in animal bodies and termed this as animal electricity. Later on, Alessandro Volta disproved this theory and stated that this was due to metallic electricity. that is, charge passing between two dissimilar metals due to electrochemical potential difference. So the correct answer is option D. Human red blood cells cannot be grown in cultures because A. RBCs require a steady oxygen supply, B. RBCs are extremely fragile, C. RBCs are terminally differentiated cells, or D. RBCs are biconcave. The requirement for culture cells is continuous proliferation. RBCs are terminally differentiated cells that have lost their ability to divide further since they have no nucleus. Thus, option C is correct. Which of the following cannot form a bilayer by itself? A. Glycerophospholipids, B. Cholesterol, C. Phosphatidic acid, or D. Glycerolipids. Cholesterol is a steroid, that is, derived lipid. It does not have a glycerol backbone with fatty acid tails. The formation of a lipid bilayer requires a glycerol backbone and saturated fatty acid tails. Thus, out of these, cholesterol cannot form a bilayer by itself. So the correct option is B. The poison dart frog or poison arrow frog in the family Dendrobatidae carry batrachotoxin among other agents. The toxin irreversibly binds to voltage-gated sodium channels and keeps them open. Why does this paralyze and kill you? A. The ionic gradient gets lost and the nerve cells die. B. Synapses remain active, so muscles cannot relax. C. The metabolic cost of pumping sodium ion exhausts the ATP reserves of the cell. Or D. All pain-conducing nerves become active simultaneously. Now, we know that batrachotoxin is an alkaloid poison. Its structure has a steroidal core. It permanently is able to block transmission of nerve signals to muscles. It binds to and opens the sodium channels and prevents them from closing. Thus, synapses remain active. So the correct answer is option B. Synapses remain active, so the muscles cannot relax. And that is why batrachotoxin can kill you. The monoamine serotonin is synthesized mainly by enterochromaffin cells in the intestine. A mouse was treated with broad range of antibiotics for a week. After a week, both the diversity of gut microbiota and the serum serotonin levels were reduced. In another independent study, a co-culture of lactobacillus and enterochromaffin cells induced serotonin synthesis. Based on these observations, the reason for decreased serum serotonin in antibiotic-treated mouse would most likely be A. Antibiotics inhibit serotonin synthesis. B. Gut microbiota play an important role in serotonin biosynthesis. C. Antibiotics make the mouse depressed and hence reduce the levels of serotonin. Or D. Antibiotics degrade serum serotonin. We know that monoamine serotonin is a vasoconstrictor. It is produced in the intestine. After antibiotic treatment, the serotonin levels were found to be reduced. Antibiotics are used to control gut flora. Therefore, we can say that the gut microbiota plays an important role in serotonin synthesis. The independent study mentioned in this question also backs this hypothesis. So, option B is correct. Konopka and Benzer isolated mutant fruit flies that had abnormal daily circadian rhythms. The graph below shows the number of fruit flies that eclose each hour of the day. Eclosion here means emergence from the pupal case. Which of these statements can be reasonably deduced from the given data? A. The wild type has 24-hour rhythm. B. The mutation in mutant 1 causes lethality. C. The rhythm is abolished in mutant 2. D. Mutant 2 and mutant 3 have mutations on different protein coding genes. From the graph, we can see that a large number of wild type flies eclose regularly after approximately 24 hours, as you can see here. Thus, the wild type has a 24-hour rhythm. So the correct option is A. Thyroxine is a major hormone produced by the thyroid gland. Rats may be thyroidectomized, that is THX, either surgically or chemically. In an experiment designed to investigate the relationship of the thyroid gland to metabolism, that is oxygen consumption and protein synthesis, following data were recorded. Experimental and control animals were tested by measuring oxygen consumption in a respirometer for 1 hour and by measuring incorporation of injected triticated leucine, that is 3H-leucine, in the liver. Post-injection, rats were sacrificed at various time points and radioactivity in the liver was measured. Based on the above description, which of the following control experiment should also be performed to confirm the relationship of thyroxine to respiration and protein synthesis? A. Inject untriticated leucine in the THX rats and take the measurements again. B. Inject untriticated leucine in normal rats and take the measurements again. C. Inject thyroxine in the THX rats and take the measurements again. Or D. Inject thyroxine in the normal rats and take the measurements again. Now, this is the given data. In normal rats with thyroid, the triticated leucine incorporation was 214 millimoles per gram per hour and the oxygen consumption was 32.6 ml per gram per hour. In THX rats, the same, that is the triticated leucine incorporation was 156 millimoles per gram per hour and oxygen consumption was 23.4 ml per gram per hour. Removal of the thyroid gland decreases protein synthesis and thereby decreases oxygen consumption. Thyroxine stimulates a series of intracellular secretory proteins at the pre-translational level. So, after injection of thyroxine in THX rats, it will increase the protein synthesis and show more triticated leucine incorporation. So, the correct answer is C. Inject thyroxine in THX rats and take the measurements again. Thank you for watching. Please like, share and subscribe to Vigyan Ghar's YouTube channel for more such videos. Next up, we shall discuss the TIFR GS 2020 question paper. Thank you.